proteins can truly be called the most versatile of the biomolecules. They perform many roles inside the body. Even organisms such as virus, which have nothing other than the genetic material, have a protein coat. These are most abundant. They form 50% of the body weight. The genetic information is mostly expressed as proteins. The building blocks of proteins are amino acids, which are joined by peptide bonds. The amino acids join to form polypeptides. The elemental composition of proteins is carbon, hydrogen, oxygen, nitrogen, and sulfur. Besides these, additional elements such as zinc, phosphorus, iron, and potassium are also present. Now, since amino acids are the units that form proteins or polypeptides, let's start with the structure of amino acids. Amino acids are building blocks of proteins. There are 20 different types of amino acids commonly found to form proteins. There are some other types too, but these 20 types form polypeptides. All of these are substituted methane, which means all of them will have one carbon, the alpha carbon. This alpha carbon has 4 valency position. Now in methane, all these are occupied by hydrogen. But here, one hydrogen is replaced by the carboxyl group, one is replaced by the amino group. So now you have an amino and an acid. But if this would be the structure, all amino acids would be alike. We have 20 amino acids. That means there is some variability in the different types of amino acids. Here we have a variable R group instead of H. Now this R can be many things, and depending on the nature of R, we have different types of amino acids. So this is the basic structure of amino acid. The physical and chemical properties of amino acids are due to these 3 groups: the amino group, the carboxyl group, and the R functional group. Now the amino group may occur as NH3+, which is basic. COH may occur as CO-, which is acidic. So the net result is neutral. The amino acid should always be neutral. Now we know that R is the one which introduces variability. So if R is H or CH3, hydrogen or methyl group, the amino acid is neutral. It has no net charge, as we just saw. There are some amino acids where other than hydrogen or methyl group, other groups may also be present. For example, serine has an OH group. But irrespective of that, if the R is neutral, the overall amino acid remains neutral. If R is acidic, which means this 2 contains a CO-, there are now more COO- than NH3+. So the amino acid becomes acidic. If R contains another amino group, then the net charge is positive. The amino acid becomes basic. So based on number of amino and carboxyl group, amino acids are acidic, basic, and neutral. Similarly, there are also aromatic amino acids such as tyrosine, phenylalanine, and tryptophan. And now a very special property of amino acids. You must have come across many compounds which have polar molecules, that is with a positive and a negative part. But these 2 types of charges are balanced and neutralize each other, and the net charge is 0. The molecule is said to be neutral. But in amino acids, the amino and the carboxyl group are ionizable at different pH. Even in a neutral aqueous solution, amino acids occur as dipolar ions. That is, ions showing both positive and negative charge simultaneously. such ions are known as Zwitter ions. Based on polarity of R groups, amino acids are divided into 4 groups. R group may be non-polar or hydrophobic. It may be polar but uncharged R group. Examples are R groups with OH or R groups with SH. Next, R groups which are polar and positively charged, and finally R groups that are polar and negatively charged. Let's see some examples of each groups. So here you have your 20 amino acids. Some amino acids cannot be made by our body and have to be supplied in our diet. These are essential amino acids, but some other can be made by our body and need not be supplied in our diet. So these are dietary or non-essential amino acids. Remember, non-essential does not mean that we don't need it. It just means we do not need to take it from outside. It can be made by our body. Let's go back to proteins or polypeptides now. As you just saw, there are 20 different types of amino acids, and these are like 26 alphabets which form many words. So peptides are polymers of amino acids, and since not all amino acids are alike, these peptides are heteropolymers. A homopolymer is one in which all monomer units are alike. You can see here that the monomer units are not alike. Different types of amino acids joined together to form the protein or the peptide unit. Now, proteins have many functions. These are the main roles that proteins perform inside our body. And now some significant proteins. The bonds linking amino acids are known as peptide bonds. So here are your two amino acids, amino acid 1 and amino acid 2. The peptide bond is formed when the carboxyl group of one amino acid reacts with the amino group of the next amino acid. The OH of carboxyl group binds with the hydrogen of amino group, and the two together are eliminated as water. One thing you may notice here is that no matter how many amino acids you join in between, one end, the beginning, will always have a free amino group and the other will always have a free COH group. So the left end with free amino group is known as the N-terminal, and the right end with free COH group is known as the C-terminal. The bond formed between the two amino acid is known as the peptide bond, and during this a molecule of water is released. Just joining the amino acids in a chain forming peptide bonds does not give us functional peptides or proteins. A peptide chain is folded into specific and precise 3D structure, and this 3D structure is required for the specific biological function of protein. So how is this chain of amino acid converted into a 3D shape? This can be understood by a 4-step sequence. Each step shows a level of structure of protein. The first level is the primary structure. This is a linear chain. The left end at N-terminal is the first amino acid, and the right end, the C-terminal, represents the last amino acid of this chain. The protein thread is now folded. This can be either in the form of a helix, like a revolving staircase, or a beta-pleated sheet. This folding is known as the secondary structure, and it is visible like this. Now the long protein chain is also folded upon itself like a hollow ball of wool. This gives rise to the tertiary structure. This is the three-dimensional view of protein. Tertiary structure is absolutely necessary for the many biological activities of proteins. Here you can see the primary structure or the sequence of amino acids joined together by peptide bonds. Then the secondary structure may be the alpha helix or the beta pleated sheet where the chain is folded, and then the tertiary structure where the alpha helix or the beta pleated sheet are folded upon itself to form a ball-like structure. Some proteins are made of more than one polypeptide unit. Each of these polypeptides are known as subunits. These subunits are arranged in a specific manner with precise orientation in space. This specific architecture of protein is known as the quaternary structure. As you can see here, the adult hemoglobin contains 4 subunits. There are 2 alpha and 2 beta subunits. 2 are alike. And then there is the heme group. So these are the various subunits of the hemoglobin protein which are arranged in a specific manner to form a hemoglobin. This arrangement of different subunits to form the complete protein is known as the quaternary structure. So these are the 4 levels of protein structure: primary, secondary, tertiary and quaternary. Now with this we come to the end of this very important session. These are your questions for practice.